All the food you've ever eaten in your life has come from photosynthesis. Either plants grew with photosynthesis or animals ate plants that grew with photosynthesis. So for our Mars to table solution, we asked, can you grow food on Mars without photosynthesis? Here on Earth, most of our food is grown outdoors. It takes up huge amounts of space and it receives energy from the sun to perform photosynthesis. But on Mars, it's going to be very different. It's going to be very difficult to replicate this. The sunlight is less intense and the outdoors is inhospitable. You could potentially grow crops indoors in a vertical farming type system, but it turns out that plants are really energy inefficient, and this was going to take a lot of energy resources to grow crops for food production. Plants only convert about 1% of the energy found in light into their biomass. So our technology uses a more efficient way to convert electricity into food. So instead of using light to perform biological photosynthesis, we take an approach called artificial photosynthesis. With this, we take electricity, CO₂ and water, into an electrolyzer that then reduces the CO₂ into acetate and splits the water into oxygen. We can then feed that acetate to food-producing organisms like yeast, mushrooms, and algae, and plants, and cultivate them in the dark. Then we can produce food independent of photosynthesis. We have developed a whole technology stack around this approach. It first starts with the electrolyzer. Our team has developed a two-step tandem electrolyzer that holds several world records for CO₂ to acetate purity, concentration, and scale. And with this acetate, we've been able to grow algae at both small and large scales. We've also been able to grow pearl oyster and blue oyster mushrooms with acetate and successfully been able to get them to fruit. Ultimately, we want to be able to grow plants and plant cell cultures with acetate. So towards this, we've made a lot of progress in engineering plant metabolism to use acetate as a carbon source. And we've also done a lot of evolutionary studies to evolve plant cell cultures to use acetate as a carbon source. If we're going to grow plants with acetate, it makes the most sense to only grow the parts you're going to eat. So for a fruiting crop like tomato, we've been engineering the architecture of the plant to flower and fruit rapidly to produce very small amounts of inedible biomass. We've also been able to demonstrate that we can grow worms on acetate as a carbon source, so we can produce animal protein with our approach as well. Ultimately, our process is more energy efficient than traditional biological photosynthesis. This is Nolux, our integrated Mars-to-table food system for 15 crew members over 500 Sol surface mission. The layout is organized around the complete path from resources to meals. We have biological production and processing on the right side and the kitchen, dining, and crew habitat on the left. This separation supports food safety, but it also keeps the distance from harvest to preparations short. Our system is comprised of 20 vertical racks that grow mushrooms and vegetables, and 9 food culture vessels, 3 shared fermenters, and 5 plant cell culture vessels, and 1 vinegar eel system. Acetate is the backbone of our process. We have 4 8 kilogram per day acetate modules. This is where the production begins, 32 kilograms of acetate per day. They enter 1,000 liter storage tank, where it acts as a metabolic buffer. We can produce acetate during low times of electricity demand. Acetate is a great carbon molecule because many different organisms can use it, and we can make it very efficiently with electric catalysis. This is really the main advantage of Nolux. Instead of relying on slow, light-grown crops, we can give carbon to all of our food-producing organisms and grow them more quickly. A lot of our crops require similar types of nutrient medias, so we mix them together in a sterile media prep zone, where we can have consistency and make sure that the media is sterile before it goes into the fermenters. Our system has 2 large food-grade fermenters that schedule campaigns for soy, plant cell culture, egg whites, protein, culinary oil, DHA-rich microbial biomass, and edible yeast. We have a smaller set of shared fermenters that support lower volume products and seed train operations. These dedicated vessels produce orange cells, pinto bean cells, strawberry cells, and other plant cell cultures without competing with the large fermenters. This sharing equipment across the campaigns, it reduces the installed mass but also it allows us to have some flexibility. Each production route is independently controlled, and this limits the chances of contamination. If one fails, then it can go to the waste and we can restart that fermentation. We've built resiliency into this system because we have backup acetate stores and we always have some backup food in case one of the large fermenters fails. So this makes our system resilient, but we didn't duplicate every unit because we need to balance the modularity with having a system that's not so brittle. We also produce whole vegetables. And so this gives us a lot of texture, colors, and freshness to our food that fermentation alone cannot provide. So we have independently scheduled shelves that produce tomatoes, onions, green peppers, lettuce, and pearl oyster mushrooms. We also have some dwarf corn that can grow in the system that can be processed into masa and tortillas. Because we stack the modules, it uses the volume very efficiently. And we've designed it so that each shelf can run independently. So we can independently plant, harvest, or clean the shelves without taking the whole rack or room offline. So these foods are important, but also they're linked to our Nolux system. We feed them acetate, and that helps boost their growth. So we aren't dependent on light in the same way that if we were growing without a heterotrophic carbon source. But another great advantage of this is that we can redesign the plants to really produce the parts that we're going to eat. For example, our tomato plants, we primarily grow only the fruit. And this is really advantageous because we're giving them the carbon and we don't want that going to the inedible parts. We've decided to include aquaculture into our system to produce tilapia. This produces a familiar animal protein to the crew. Now, it's very resource intensive to produce this tilapia. They go through a two-step system of making fries to the adult fish, but we decided to include it because even though this is not the most energy efficient route for food production in our system, it's very deliberate because we're really focused on the quality here and the variety for the crew. We don't want the crew to be eating things that they don't want to. So we have put in special meal plan to allow them to have something to look forward to. And the aquaculture tilapia is one of those. So to process all of this food, we have a bioprocessing suite attached to the production and the kitchen. And this is where all the ingredients come to get processed. We have many different unit operations in the bioprocessing site so that we can process all of our fermented food products, the vegetables, and even the fish. This connects to the kitchen, which the ingredients flow to, and this is where the meals are created. And then after that, the kitchen goes to the most important part, the dining table. And this is where the crew will gather to enjoy their meals. So in our final integrated simulation, our Nolux system created 8,000 different production runs of different types of crops and over 9,000 unit operations across the 500 souls. And this was able to execute with no execution shortages in the meal delivery. That means if the system operated perfectly, we wouldn't have any hiccups. And we've been able to run several simulations that show our system is very robust in delivering the required nutrients to the astronauts.
Ultimately, this closed-loop system allows us to produce a very wide variety of meals with very simple inputs. And some of these ingredients can be upgraded into other ingredients that really go towards improving the nutrition and quality of the food that these astronauts will be consuming. For instance, coastal Mexican tilapia. For lunch and dinner on Sol 7 and 13, there's 5-spice tofu and mushroom corn fritter lunch. For dinner on Sol 7, there's Italian mushroom flatbread. And breakfast on Sol 8, there's French toast with a strawberry plant cell culture compote. Ultimately, these are some of the exciting dishes that we can produce with this system. And there's a much broader list as well, all using primarily ingredients grown on Mars, with small substitutions to enhance what we could make that's brought along. These base ingredients make up their calorie needs and nutritional needs of the astronauts, and we are tailoring it based on their consumption, whether they're going on EVs or activity, to really optimize calorie and nutrient requirements so that we're not overproducing or underproducing for given people and avoiding just an average calorie content. And ultimately, our system can recycle a lot of waste products and really turn them into a wide variety of delicious food for astronauts exploring Mars.
Using the Nolux approach, 68% of the calories consumed by the crew can be produced on Mars. And we can do it with only 2 crew members, enabling the success of the entire mission. We are Nolux.